All right. So welcome everyone. Welcome to the last webcast in the Leukemia and Lymphoma Society of Canada's Innovation and Research Series, Precision Medicine, Finding the Right Treatment and Improving Treatments for Each of Us. Scientists continue to gain a deeper understanding of the genetic changes that lead to the development of cancers. This knowledge is being used to develop more precise treatments for blood cancers. And in today's presentation, we have Dr. Aly Karsan, who will speak about the latest research developments in precision medicine. My name is Natasha Sani, and I'm the Community Services Lead for the BC Yukon region, and I'll be the host for today's online event. Since there are many of you who are here in the audience, we invite you to type your questions and comments in the question box of your Zoom webinar panel throughout the presentation. An LAHSC staff member will monitor the questions. At the end, I will read some of your questions aloud during the question and answer period. Please keep in mind that since we have such a large audience today, we may not be able to answer all your questions. And also note that Dr. Carson will not be answering personal health questions or questions that may not be related to the topic. But please know that if we do not get to your question during our live presentation today, a community service lead will contact you after the webinar to assist you. Today's presentation is recorded so you can listen to it again on our website. Before we begin our main presentation, I'd like to share our mission and highlight some resources that might be helpful for you or someone you know. Next slide, please. At the LAHSC, our mission is to cure blood cancers and improve the quality of life for those touched by blood cancer and their families. We listen attentively and with compassion to the challenges you are facing. We can point you to information specific to your type of blood cancer or treatment. We help you get organized to manage and take charge of your care, and we can identify services that would be beneficial for you. So please don't hesitate to contact the community services lead in your area to learn more. Next slide, please. Our website, bloodcancers.ca, was designed with you in mind, and this is to ensure easy and personalized navigation no matter your current circumstances. Next slide, please. On our website, you will find fact sheets and booklets, animated educational videos, webcast recordings, podcasts, and much more. Please visit www.bloodcancers.ca to access any of these tools. Next slide. Another resource that can help you is our LAHSC Health Manager app. This is a free app that can help you track side effects, medication administration, food, and hydration. The app also offers medication reminders and has a nutrition meal planner along with many recipes to help you get the nutrition you need. The app also allows access for a caregiver and is free to download in the App Store on Apple or on Android devices. Our virtual support groups from coast to coast are professionally facilitated and offer a safe space to share experiences and to inspire and encourage others also affected by blood cancer and their caregivers. Please visit our website, bloodcancers.ca, to learn more and to join. Next slide. Our peer support program, this is a support service that matches people affected by blood cancer, so caregivers and their families, with trained volunteers who have been touched firsthand by blood cancer and shared a similar experience. Whenever possible, participants and volunteers are matched based on their age, gender, and diagnosis, or the issue that is most concerning. Contact a community services lead in your region to learn more about this program. Next slide, please. So it's now time to introduce our speaker today, Dr. Aly Karsan. Dr. Carson is a hematopathologist at the BC Cancer Agency and Tier 1 Canada Research Chair in Blood Cancers. As a clinician scientist, Dr. Carson's dual roles in the clinic and his translational research program in AML and MDS feed on each other to drive progress in understanding mechanisms of therapy resistance and relapse in myeloid cancers. Thank you for being here today, Dr. Carson.
Thank you. Thank you, Natasha, and thank you all for joining. I have to say I'm in Vancouver, but it's unseasonably hot. So if you see me sweating, it's not because of your difficult questions. I'm just trying to get used to the heat. So I was asked, and I want to thank the Leukemia and Lymphoma Society of Canada both for asking me to speak here, but also for funding some of our research. So today I was asked to discuss precision medicine and about the work we're doing in the area. But as I tried to explain some of our work, I realized that we need to start on the same page. And so for you to really understand what I'm talking about, I need to give some background. And I apologize to those of you who know what DNA is and what cells are, but that's where I'm going to start. And so this first slide actually shows in the background a bone marrow. So it's a biopsy of a bone marrow, and it's taken from a real patient. And the way we do it is we take a biopsy from the hip bone and then stain it. And what you see here are all the types of blood cells that are produced in the bone marrow. So you see what will eventually become a red cell that actually you've probably seen and looks something more like this over here, but they start out like that. There are white blood cells that look like these over here. And then there's platelets, which are little cells that come from a bigger cell called a megakaryocyte. But there's also other things. There's fat cells, and there's, if you follow my arrow, the outline here is a blood vessel. And blood vessels are really quite important because the proximity of blood stem cells, the cells that make all the blood, to the blood vessel is very important for those stem cells to be maintained. So how do we get to, and I should say, I'm going to focus most of my conversation around acute myeloid leukemia and some other myeloid cancers, because that's what our research is. And so the first question is, how do you actually get to an acute myeloid leukemia? So this on your left is all the bone marrow, all the different types of cells you have in the bone marrow, and there are many different kinds of blood cells. At some point, a mutation will occur, a change to your DNA, and that gives one cell an advantage. And so that cell starts to expand and form something called clonal hematopoiesis. Now, clonal hematopoiesis in itself is not leukemia. It just means these cells have some sort of advantage. They have an advantage over all the other cells. But then over time, there are more changes to your DNA, and that leads to different types of cells coming out. So one cell might get one change, ends up looking like this, but another cell might get a different change, ends up looking like this. And then somewhere in between, as that cell is dividing, a third change might occur that gives you even a third set of genetic clones. So even when a person is diagnosed with cancer, this is true of probably all cancers, it's not just one type of genetic defect. There are multiple genetic defects in the same patient. So when you have the same type of genetic defects in a bunch of cells, we call that a clone, because they all came from originally one cell. And what you can see here, I'm showing you is 3 different clones. So already it's very complex, even before you're diagnosed. Then,
sorry to interrupt. We just had a request, if possible, if your pointer can be enlarged for the presentation. I don't know.
I can't enlarge it. Let me see if I can try something different, though. I don't know if this is this better?
Oh, perfect. Thank
Okay.
you.
So what happens then after diagnosis is you get chemotherapy, and that's called induction chemotherapy. So when you get therapy, you may be unlucky and the cells don't respond, or maybe some cells respond. These 2 clones respond, and what grows out is these cells here because they are resistant to the therapy. But most people will get into what's called remission. Now, remission is not the same as a cure. It just means that there are not enough cells for us to see when we look down the microscope. And at the time of remission, it might be that you just have those 3 different types of genetic clones, but it might also be that during the time you're being treated, that another mutation has happened, so you've got an extra clone now. So it's getting even more complicated. And to really try and achieve a cure, what you can get either consolidation chemotherapy or a hematopoietic cell transplant, and that's really dependent on how aggressive your disease is, what you'll get. Now, you may be cured here, but if you're unlucky, then you get a relapse, and those cells come back. So how can precision medicine help along this journey? So the things that we're currently doing and is at diagnosis. The first thing we do is we do a genetic or genomic test. So we sequence your DNA, we look for all the changes in the mutations. And the one thing it does is it helps us say, are you going to be in this group that's more likely to be cured, or are you going to be in this group that's more likely to relapse? But, you know, so that's helpful information because it helps decide how aggressive treatment you should receive. Should you receive more chemotherapy or a hematopoietic cell transplant? The other thing, though, it can do is it can help choose different therapies. So until a few years ago, I would have said, you know, the change in treatment for AML has been zero in the last 30, 40 years. But over the last few years, there have been a number of therapies now that are targeting specific genetic mutations. And so by sequencing your DNA, we can decide what kind of therapy to give you. But we can also look at the time of remission, where down the microscope, we don't see your cells. We can start testing and saying, okay, if we test your DNA and look for specific mutations, how much of your AML is remaining? Have you been really, have we really killed a lot of cells, or have we not? And then this can be monitored. And that also helps in decision-making about which way to go for therapy. So just to put this sort of in a video, you start out, this is your an example of a bone marrow that I drew. These are the blood vessels where the stem cells are close to, because they need to be close to in order to survive and grow. At some point in time, you might get a mutation. In fact, we're all going to get mutations in some cells in our bodies, every single one of us. And I'll talk more about that later. But that cell, when you get that mutation, it might give your stem cell an advantage, so it starts to expand. So it makes another stem cell, but it's also making other cells that are actually blood cells, the same blood cells that we normally use. And so you get more and more of these, but slowly what's going to happen is these things are abnormal because they have a mutation. They start to secrete chemicals. So these cells start to secrete chemicals, and those chemicals start to block normal stem cells. So now this stem cell, which has a mutation, has an advantage, so it can grow, can take over the space of that other stem cell. Now suddenly you're getting what I showed you before, what is called clonal hematopoiesis. And you get more and more stem cells, and then, you know, eventually one of those is going to go to another site to another bone in the marrow where the blood is growing, and that's going to expand there. And eventually, or possibly for some people who are unlucky, you'll get another mutation, and that'll give you a cancer. In this case, leukemia or a myelodysplastic syndrome or something else. Okay, so what exactly is a cell? I've been talking about cells, and every tissue in our body, every organ is made up of different cell types. So we can have nerve cells or blood cells, muscle cells, bone cells, fat cells. Every one of those things are cells. You can also have, you know, sperm and ovaries, which are called germ cells. And all of these different cells come from the mother of all cells, the stem cell. And we'll talk about that later. But this one type of cell, as you grow, and even as an adult, we have blood stem cells that make all the different kinds of blood cells. These cells can make themselves, and they can make the blood cells that we need to function. So what is within a cell? And you can take so this is an electron micrograph. So it's a microscope that uses electrons to interrogate the cell. And you can see that this cell has a membrane around it. So you can think of it as, the analogy would be, you can think of it as a factory. It has an outer membrane. Within it has these things called mitochondria, which generate energy. There are things called lysosomes. There are waste disposal stations. There's a place to make more cell components and called the rough endoplasmic reticulum. So you make all the different components of the cell there, and some of these are modified in another part of the body. What I want to talk to you about, though, is the nucleus. What is the nucleus? The nucleus houses all your genetic material right there. And so that's where your DNA is found. And DNA, you might hear the term base pair, is formed of two strands. So there's one strand here, you can see, and another strand here. And there's only 4 letters that make up all the blueprint of how that factory what that factory is going to do. And those 4 letters, the way they're arranged, gives you a sequence. Now, those letters are actually chemicals, and they form bonds with each other. So on this strand, if you have an A, it always pairs with a T, as you see here, the blue and the green. If the A is on this strand, it'll still pair with a T, but on the other strand. And the same happens with the G and the C. And so what you have are these 2 strands, which then get coiled into a DNA double helix, for which Watson and Crick are famous for describing this structure. But that's not enough, because this DNA, from a single cell, if you stretch it out end to end, it's actually 6 feet long. It would be 6 feet long, and somehow you have to get that 6 feet of DNA into the nucleus, which is about the 10th of a slice of hair. So if you slice your hair into 10 parts, that one lengthwise, then one of those would be about the size of your nucleus, and 6 feet of DNA needs to fit in that. And so it gets coiled. It gets coiled around proteins. We'll talk a little bit about what proteins are, but then it gets coiled even more and more until at the end, it gets wound so tightly that you end up with these things called chromosomes, and they're found in the nucleus. So precision medicine, so the definition that the NIH uses is, it says that it uses specific information about a person's tumor. Now that specific information is usually what that DNA sequence is that you have, or what are the mutations or the changes in that letter code that have happened in your cancer. And we use that information to help us make a diagnosis, to make a prognosis, in other words, to say whether you're going to do better or worse, and it helps to plan treatment depending on what that shows, or if there's a mutation for which we know there's a drug that will target that. And we also use it to find out how well treatment is working. So what are these genetic changes that I'm talking about? And you can break that down into the big stuff, which I'm showing you here. So the big stuff are big changes in lots of DNA, so much so that you can actually look down a microscope and see changes in the chromosome. So the kinds of things you can get is, for instance, a deletion. And a deletion on chromosome 5 is a common thing that you see in chromosomes from myelodysplastic syndromes, so that you have a smaller chromosome and you've lost all this genetic material on one of your chromosomes. You could also get an insertion so that you get a big piece of DNA inserted into different areas. The other thing is you can get an amplification. So an amplification is different from an insertion because really it's taking one part. So it could be a gene or even bigger than a gene, and that gets duplicated or it gets even further enhanced. So there, an example of this is seen in breast cancer, where some of you may have heard of HER2 positivity in breast cancer. And that particular amplification means there's more of this protein, so it drives the cell to keep dividing, but there's a drug that targets that particular gene called Herceptin. And I should say, if you have a deletion of chromosome 5 called del5q, that can be treated with lenalidomide, and we'll get to that later. And then finally, the third type of big change you can get is when one part of one chromosome, in this case chromosome 9, moves to chromosome 22 in this case, and the part on chromosome 22 moves to chromosome 9. And then you end up with this smaller chromosome, which is a combination of 9 and 22, and then another chromosome that has the remaining material. But this particular mutation puts two genes beside, or parts of two genes beside each other, called BCR-ABL. And BCR-ABL is what actually defines one of the blood cancers called chronic myeloid leukemia. Luckily, there's a treatment for that called imatinib. And in fact, it's so successful that most people do not need anything else. This was the sort of wonder explanation or wonder drug that happened in the 90s and early 2000s. And then there are other translocations, similar examples, similar repercussions in determining who should get treated or what they should get treated with. But there's also other things. There are smaller mutations where the code is changed so that only one letter changes. So in this case, a T gets mutated to a G. And that has a big impact when it happens in this gene called JAK2. That ends up in giving you a different kind of protein, which causes myeloproliferative neoplasms. You can have insertions or deletions of small things right here. So here you can see an insertion of 4 letters, but that's enough to cause a protein change that results in AML or makes AML progress. But it also means that you actually have a better prognosis than if you don't have this mutation. And then similarly, you can have a duplication where a small piece of the letter code here is duplicated so that now you have the sequence doubled up here, what's called a tandem duplication. So to try to see what's happening here with these small mutations, you have to do what's called molecular genetic testing or DNA sequencing. Whereas here, when you're looking for big things, you do something called cytogenetic testing, and now there's new types of equipment, and we refer to it more as cytogenomic testing. Regardless, what we're looking for are these different things, the big things and the little things that change specific letter codes. Now, the other thing we need to talk about, and this can be quite confusing for a lot of people, is germline versus somatic mutations. So germline mutations are DNA alterations that are already present in all your cells at birth. And usually that's because either one of your parents has that mutation already and they pass it down to you. Somatic mutations, on the other hand, are acquired. They are new DNA alterations that occur in a specific cell or tissue. And how do they happen? Well, every time a cell divides, the machinery that makes the new copies of your DNA makes mistakes. It makes lots of mistakes. Most of them are actually repaired. But every single time one cell divides into 2 cells, you acquire 1 or 2 new mutations. So you can see that even as we're sitting here talking, your body, there are cells that are dividing and they're acquiring new mutations. And so, it's a wonder that we all don't end up with cancer when we're 10 years old. But our body has many ways of protecting us against that. So even a newborn, if you look, will have 50 new somatic mutations that are not found in either parent, that have occurred because as soon as the cells start dividing to make the fetus, and then, when the baby's born, will give you new mutations. So I already said that. But so when we talk about germline mutations, we can say that about 10 to 15% of cancers, and this is true for blood cancers as well, are familial. In other words, people have inherited a gene mutation that makes them more susceptible to developing cancer. And sometimes those mutations make you more susceptible to multiple different kinds of cancers. Sometimes it might be focused on a single type of cancer. And as I said, the risk for development of cancer begins before birth because each human is born with many mutations. And if you keep going, a 100-year-old person will contain 2,000 to 4,000 mutations in a blood cell. So that's just looking at blood cells. And it's been found most recently that mutations that drive leukemia can be found decades before cancer develops. So, this having a single or even a couple of changes in your DNA doesn't mean you're destined to get cancer. So what is the scale of a whole genome? And a whole genome is all the DNA that you have in your cell. What are we talking about? So there are 3 billion base pairs. So that's 6 billion bases, right? 2 strands, and they're different sequences, even though they're kind of complementary, right? So here's the letter code on one strand. The opposite strand will have a different letter code, but they match. So the analogy here is if you took one base and that was 1 second, we would have the equivalent of 180 years of DNA letters in one cell. That's a lot of seconds, and that's how much DNA we have in a cell. Now we have between 500 billion, 500 billion to 1 trillion blood cells that are made a day. And if you had with 1 cell or 1 second, that would equal 15 to 30,000 years. So we have a lot of DNA in our body. And so, now we're talking about trying to look at the whole genome of an individual, because our current testing, for the most part, like that's in the clinic, is looking at, a whole bunch of genes in the range of between 50 to 500, depending, for things that we know are important in cancer. But it's also estimated that we only know about half the genes that are important in cancer, and there's many other genes that we just haven't learned about yet. So, how are we going to solve this problem? And that answer came around 20 years ago. So when the first human genome was sequenced, it took 13 years, about $3 billion US dollars, to get one sequence of one whole genome. In 2005, a new technique called next generation sequencing was introduced. It's also called massively parallel sequencing, because instead of sequencing one little bit of DNA at a time, you end up sequencing millions of bits of DNA at the same time. So now, routinely, within 2 days, you can sequence a whole genome. But there's a problem, and the problem is this. So the cost of sequencing, here's the price, and along here is time. The cost of sequencing after the introduction of next generation sequencing with new innovations and new machinery, the price started coming down to the point that we're around at $1,000 US to sequence a whole genome. The problem is now we have 6 billion letters to look at. And obviously nobody can look at that one by one. So we have to use computational methods. But that still takes a lot of time. And the cost of computation, even though this is Moore's law for those of you who know computing, which says basically that the price of computing will drop at an exponential level over time, and it keeps dropping, but it's not dropped as fast as the cost of actually sequencing. So we've got all this data, and it's just impossible to analyze all of it completely and thoroughly and completely accurately. So if you want to do whole genome sequencing, those are the caveats. But nevertheless, we can do it. We have been collecting lots of whole genomes across the world, and now it's going to take many years to try and really understand what's going on. So, if you look at Wikipedia, it'll tell you the Human Genome Project was completed in 2003. If you look at the National Institutes of Health, who actually funded a large part of the sequencing effort, they'll say, well, the final complete human genome was described in April 2022. Now, that's 20 years difference here. And the difference is that we could sequence about 90 to 95% of the genome, but there were very difficult areas to sequence. So it took 20 more years to actually get through and sequence all those little parts. So as I think many of you will know, it does take 90% of your time to address 10% of the problems that you have, or even less. So this is exactly the case that happened with sequence. But then if you look at the BBC in 2023, it says why the human genome was never completed. And what they were talking about is why it took 20 years to get here. But also, the human genome is not complete, and it will never be complete. And the reason it will never be complete is that we're all different. So we all have different genomes, and that's what makes precision medicine possible, is looking for those differences. So even though, any individual on the planet has about 99.6% of their DNA identical to any other individual on the planet, 0.4% represents about 12 million base pairs, so 24 million bases. So lots of places that are different that make us, each individual, what they are. And so, changes in either those regions or even in common regions is what makes every tumor a little different from one person to another. So, when we look at genetic and clinical factors that influence outcomes in AML, we can see that we break people down into three areas of risk, or three layers of risk. There are the patients who are of favorable risk, but still only 50% of those survive. So this gives you the probability of survival, and this is time. So over time, you'll see that at around a year, or let's say 2 years, 50% of people will survive if they're in a favorable risk category. And that's about 30% of people with AML. In the intermediate risk category, this is about 25% of people. And then when you get down to the poor risk, which is another 30% of people, it's a very, very low number that will actually do well. And so, as I was saying to you a little bit earlier, if we use DNA sequencing or other kinds of molecular methods, we can find some of the big abnormalities and some of the little abnormalities that tell us which category to put each individual. And those different categories determine, as I was saying before, what type of therapy you should get. Now, there's one problem, as you see here, is the intermediate category, where it's really, it becomes very dependent on other factors, which way to go. Do we go this way? Do we go this way? Do we go with a different kind of stem cell transplant? And it gets quite difficult. So really, one of the goals is to take the people who are in this category, and we can see that there are people who do very poorly. So about 50% of people in the intermediate risk category will die very early on. But then these 25% of people survive. So they really are, these people aren't intermediate risk. They should be piled up here with the good risk people. And those people that die are also not intermediate risk. They're poorest. They should be here. So how can we improve this so that we can get these people in the middle to the right group so we know best how to treat them? So one way is with the same sequencing method. When we sequence all these genes and look for all the mutations and DNA alterations that there are, there's one mutation, for instance, FLT3-ITD, which actually is not, it's not good to have, or I should say it was not good to have. But now there are inhibitors, there are drugs that actually target this mutation. So you would be eligible for additional specific therapy that targets this particular genetic mutation. Now, there are other mutations that are not part of your prognosis classification, so they don't really impact on whether you do well or not, but you can still target them because they're important in driving the leukemia. And these are called IDH1 or IDH2, and there are inhibitors against those genes or proteins. And there are other drugs that work that don't target mutations, but for instance, venetoclax targets a protein called BCL2, which helps the cell survive. And it's not that there's a mutation, but for some reason that there's more of that protein in some people, so that they do well when you give them this particular inhibitor. And then there's other drugs where they work, but we just we don't know. We still don't know. Now, 5-azacitidine has been around for decades. It's been used for a couple of decades in myeloid cancers, but we still don't know what this drug is targeting, even though we can see that it works. So as you can see, there's still a lot to learn. So I showed you this slide before, and I want to bring it back because I said this one stem cell can make all these different kinds of cells. So the obvious question is, if one individual has the same type of DNA in every single one of their cells, there might be a few mutations in one or the other, but that's not what's making the blood cells and nervous cell or the nerve cell. So how is it with the same blueprint, you get a different cell, a nerve cell, which is quite different from a muscle cell or a bone cell? So let's see. There are about 200 different types of cells in the body, and even in the blood system, we talked about the big types of blood cells, the white cells, the red cells, and the platelets. The white cells fight infection, the red cells carry oxygen, the platelets block bleeding, so they clot your blood. But there's many, many different types of cells. And the reason we can get that, so to go back to the blueprint analogy, every cell has the same blueprint, but it has the blueprint not to make one factory, but to make multiple factories. And so you can make a car factory, or you could make a factory that produces furniture from the same blueprint. But obviously you can't use all that information. So what happens with your DNA is that only certain parts are revealed for that cell to make the things that it needs to make. And so the rest of the DNA remains coiled up. You remember I showed you how it was all tightly wound up. And then the other parts of the DNA that are needed for the car factory open up. And when they open up, it allows a process called transcription, which forms what's called RNA. RNA is very similar to DNA, but it's only one strand. It's a single strand. And what it does is it allows another process called translation to make amino acids. So every 3 letters codes for 1 amino acids, and we have 22 amino acids. And those things in their special sequence will fold and form proteins. And so what are proteins? Well, proteins will make, there are 2 big classes of proteins. They have different functions. There's a structural protein, so in a factory would be the beams, the roof, the things that keep the cell integrity. So if there's a cell down here, things that make up these fibers that hold up the cell, there's other fibers along the edge, the walls. And then there are the enzymes, and the enzymes are like catalysts. They speed up chemical reactions, and they allow the other things like your mitochondria, the energy producing parts of your cell, to do that quicker than if you just left them around. It would be like cooking with a stove as opposed to trying to heat something under the sun. So the enzymes have function, they help process chemical reactions, and then there's the proteins that just give the cell structure. And either one of these things going wrong can affect how your cell functions and can be responsible for cancer. So when we look at all the DNA in the cell, we call it genomics. If we look at all of the RNA, that's called transcriptomics. So we're looking at the transcriptome. And you could also look at, right now, there are way more proteins than there are genes. So it's not possible in one sitting to look at every single protein in a cell. But looking at many proteins at the same time is called proteomics. And we talked mostly about genomics, in fact, only about genomics so far. And I'm not going to talk about proteomics in the context of precision medicine, but let me tell you a little bit about RNA. And the question that we asked is, since looking at those genetic mutations still gives us those people in the intermediate category, there must be something else going on that we're not able to interrogate that tells us that this person's going to do worse, that this person's going to do better. And so we asked the question, can we look at the RNA? Can we look and see what RNAs are present in those leukemic cells? And can we see whether some RNAs are more abundant in some types of leukemia or less abundant in other types of leukemia? And can we make sense of that? And so we did that to try and improve deciding who should get what treatment. And so the first thing we did many years ago now is to develop a myeloid genomics panel where we only did DNA sequencing. When we realized that wasn't enough, we started doing RNA sequencing. And what we found was actually we could make a prognostic score, a score that told us, you know, if you have these RNAs in abundance or these ones in lower abundance together, we can tell you whether you're going to do worse or better. And if we combine this along with the DNA, then we actually do pretty well. Because if we look, we see we have 64 people in the intermediate risk category here. But when we include RNA sequencing, we're down to 29. We managed to move people either into a lower risk group along these people over here, or other people to a higher risk group. In fact, we could even take people who we thought were in a higher risk group and put them into a lower risk group or vice versa. So this test now has allowed us to shrink those people in this intermediate risk category. But it does more than that. What we found is that those patients with poor risk, and this is a very confusing slide, I know there's a lot of information, but if you'll just look at just what I'm pointing out, you can see we looked at different cohorts of RNA sequencing, including our own different groups. And what we found was the integrin signaling was activated in those poor, in a certain set of poor risk people. And in particular, one gene called PTK2 was highly expressed in a subset of patients who did very poorly. Now, PTK2 is involved in helping cells stick together. That's what integrins do, and PTK2 sends the signals that do that, so that cells stick to cells or cells stick to the underlying soil of your bone marrow, and that makes, and we don't know why, but that makes for a worse outcome. So that was something new, but we decided to look at that a little more, and we said, okay, so those people over here on the left, so each line up here is an individual person. Those people with high levels of this PTK2 RNA, if you compare to their DNA and said, what DNA mutations do these patients have? We saw that most frequently these patients had RUNX1 and TP53 mutations. These are two different genes. But they were less likely to have this FLT3 mutation, which I told you there was a targeted therapy for. So these diseases are different from the ones that have FLT3 ITD. And we saw this across different patient groups. And so we said, well, people had made PTK2 inhibitors, not for AML, because this was new, we had just discovered that, but they had made them for solid tumors, and people were using them because there was some evidence that integrins were important in solid tumors, because they're all stuck together, and so the feeling was if you could separate them out, then you would disrupt their survival. And so we tried two different ones of these inhibitors, and we said, okay, if we take cells where we mutate this RUNX1 protein, what happens? And what we found was that we could kill those cells if they had this mutation, but if they didn't have the mutation, these two PTK2 inhibitors didn't do much. They did a little bit, but really when you lose this function of this gene through a mutation or otherwise, those cells get killed very easily. And the same thing for P53 mutations. So this work, we've done some other work with it and shown some other ways that this can be regulated. And in fact, there's a group in Australia carrying out a clinical trial with a PTK2 inhibitor in AML, and they are interested in using our data as a biomarker or something to be able to say which patients are going to do better and which patients are going to do worse with this particular inhibitor. So then the question the other question that that my lab asks is, why does therapy fail, right? So I showed you this before. so remember I said, oh, this is a genetic clone. They all have the same DNA mutation in these cells, but yet I color them differently. So there's some shading here. Why the shading? Well, because there's different, even though they're same genetically, these cells may have progressed to become slightly different without changing their DNA, but changing how their DNA is packed. So there may be other areas that have opened up that were closed before in some of these other cells. What does this exactly mean? So I talked about genomics, where you have the DNA that makes RNA, right? But if you can change different areas where how the DNA is modified, that's called epigenomics. So what's happening here, you can see, is that there's a change in what we call methylation. It's just a chemical addition to the DNA letters. It doesn't change the letter code, but it changes how this chromatin, the big parts, are open or closed and how they work. And so we started looking a little bit at that. And let me just go back to sort of remember how blood cells are made. So they start with blood stem cells, which can self-renew or make other blood stem cells, or they can differentiate. They can make the red blood cells and the white cells and the megakaryocytes. Now, leukemia also has stem cells because you have to propagate the leukemia. You have to keep self-renewing, but you also get some of these cells becoming more like generally white blood cells. But what we found is that the drug called lenalidomide, which is used in patients with myelodysplastic syndrome with the deletion of that chromosome 5, and it works very well, we found that what this lenalidomide does is it makes these stem cells megakaryocytes instead of myeloid cells. And it's only these megakaryocytes that die. So there's a whole bunch of papers that were published that led up to this and actually describe these. And I'll very quickly go over this. This might be confusing, but I'm happy to answer questions after. So this is a kind of myelodysplastic syndrome with a deletion of chromosome 5 or part of it. You give the drug lenalidomide, it makes the cells megakaryocytes. How does it do it? It does it by degrading a protein called IKZF1, which allows these cells to become megakaryocytes. And then it degrades another protein in these megakaryocytes, which then results in cell death. This mechanism only works in these cells. And the reason is that this same gene, casein kinase 1, is lost in this deletion. And so when you target it, now you don't have this gene anymore, which is essential for that cell to survive. And so you kill the cell. But we also found that if you have mutations in p53 or RUNX1, that blocks the megakaryocyte differentiation. So you don't get that anymore. But remember, I just told you that we can use BTK2 inhibitors. So that's something that people might consider in the future, but these drugs in myeloid cancer are still in clinical trials. So we were interested, they've not been approved for use. We were interested in drugs that were already available and could be used clinically. And so we looked at another gene that we had described a number of years ago, also being lost in this area. And we said, well, if you lose this gene, does the cell actually change in a way that would make it something that we could target with the therapy? And in fact, we found this protein called IGF-1R gets activated when you lose this gene. And not only is this protein activated, but downstream of that protein are two other proteins, MEK1 and ABL1, which are enzymes. And there are drugs that are approved clinically. And so we asked, well, if we use those drugs, can we kill the cells? And of course, I wouldn't be telling you if it didn't, but in fact, that's exactly what happens, is if we block any of those things, IGF-1R, MEK1, or ABL1, those cells now can be killed, even though they still have those mutations and lenalidomide doesn't work. We can still do this. So now we're in the process of talking about trying to give patients who get resistant, putting them into a clinical trial. Now, it'll be a while before that happens because it takes a while to get these things set up. Separately, looking at another drug, what we found was that even at the time of diagnosis, just looking at the epigenomics, looking at how the methylation of the DNA was, or how the chromatin was wound up, or was it open, that we could look and show that people who didn't respond to a drug, specifically 5-azacitidine, had one type of chromatin. It had nothing to do with the DNA letter code, but just the way it was open or closed. The non-people who didn't respond to the drug looked one way, and people who did respond looked completely different. Their cells had a different epigenome. And so finally, I just want to say, how can we, we can find all these things, but it's not enough to just see it and make an association. We have to show that this is causal, that this change actually has an effect of making someone resistant to a drug, or making it so that a leukemia will develop, or making it so that you don't respond to chemotherapy. And so my lab does a lot of functional studies. I sort of described in the last slides a little bit of that. But, traditionally this work was done with mouse leukemias. You made a mouse leukemia and you studied. And then people had the idea, and John Dick talked a couple of months ago about this, where they put human cells into immunodeficient mice so you could grow them and look and see how that worked. And we do a lot of that. But, we're talking about human cells. So this is normal. This is, a human leukemia. And what we would like to see is these human leukemia cells mixed with human cells, the blood vessel cells that we said were important, not the mouse cells, but human cells. And so how can we do that? And so in the last slide, what I'm going to show you here is we're using stem cells, which can make every other cell type. And we're making blood cells, and we're making in green, and we're making the endothelial or the blood vessel cells, which we show here in red, and there's other cells in here which we're not marking, but you're seeing the yellow. And this yellow is where the blood stem cell is close to the blood vessel. And so we know that these are probably, or some of these are stem cells that will produce all these other blood cells in the middle. I'm just going to show a little video here, if I can find the thing. And as this turns, this is gonna, like we're on edge now, but as it turns, I just want to show you, that this yellow, you can see that even internally, there's these blood vessels where the stem cells are close to. And what we can do now is put leukemic cells into this human environment and now start to study how drugs work and what makes these things resist and what makes them survive. So in the end, I just want to go over one of the early slides and say, well, where do we go from here? So we talked about genomic testing at diagnosis and how that helps us. And we talked about even in remission, knowing how much disease was there was important. And now where we want to go is looking at people who don't have cancer yet. Is there a way we can find which of these people that have clonal hematopoiesis, which ones of them are going to actually go on to get a leukemia? Because most of them will not. The vast majority will not. So we have to find those people that actually will progress. So that's a really important thing. And then the other thing that there's already new techniques going on with is trying to develop even better assays to say, what's happening here in remission where we think we've killed all the cells, but we know we haven't, that allows some people to be cured, but most people or many people to relapse. And that's what we really want to know. Which person with clonal hematopoiesis will progress to leukemia? How can we tell which person will be cured and who will relapse? Can we find new proteins that we should be targeting? Can we change the epigenomic state of the cell to make them sensitive to therapy? So that's the directions that we are going, and other groups are going to try and unravel this really complicated system. So I'll stop there. Thank you very much for listening. I want to acknowledge all the people in my lab who did all the work, people previously in the lab. We have many collaborators, both locally, across the country and across the world, and various funding agencies, including the Leukemia and Lymphoma Society of Canada, and various other funding agencies that have supported this work. This is my lab here during the time of COVID, and here's them unmasked. So thank you very much. And I'm happy to take questions.
Thank you, Dr. Carson, for sharing your expertise and insight with us today. It was super informative. We'll now move on to our question and answer period. So if you haven't done so already, be sure to use our Q&A box in your Zoom webinar panel to type your questions. Just a quick reminder that Dr. Carson cannot answer specific questions about health status or questions not related to today's topic. These questions won't be read aloud during our question answer period, but we will have a community service lead contact you after the webinar to help answer any questions. I also wanted to note there were quite a few questions about causes of blood cancer. We will pop in a video from a previous webcast we had, the causes of blood cancer, what we know, innovation and research webcast with Dr. John Dick. So you can definitely go and view that webcast recording as well. So Dr. Carson, one of our first questions here, will advances also be able to target the genetic profile of patients with chronic leukemia, lymphoma, and myeloma, etc.?
So I was giving you the example of AML, but certainly we are working with others who are really expert in the lymphoma field to develop similar tests. Myeloma is actually one of the success stories. They've been looking at the big stuff, so the cytogenetics or cytogenomics, DNA aberrations, so things like translocations and what's amplified or what's deleted. And in fact, there have been incredible advances in myeloma over the years. When I was training, my mentors told me, myeloma is a 2-year survival. Nobody survives more than 2 years. And now people go on for a couple of decades. There's lots of targeted therapies. The testing helps tell us early on who's going to do worse, and they get sort of more treatment, continued treatment to help them along. And so, this is not unique to AML. It can be applied to any blood cancer. Not all of them have the same level of advances. AML, we particularly work on it because as I showed you, the survival is so poor compared to some of the other cancers. But solid tumors also are using these same types of technologies.
Okay, thank you. Do you believe in a definitive cure?
That's like asking me, well, I think more and more, so certainly there are definitive cures in different types of leukemia, different types of cancers. There already are. If the question is directed specifically to AML, I'd quote Yogi Berra. It's very difficult to predict things, especially about the future. But there is hope. As I said, 5-10 years ago, I would have said, well, there's been no change. There have been no new treatments for acute myeloid leukemias or myelodysplastic syndromes for 30 to 40 years. And now there's new treatments, and people, it appears, are living longer. And whether by cure you mean completely eliminate every cancer cell, I certainly believe it, and certainly that's what we're trying to head for. But it's been a very complicated and complex task because we thought sequencing the genome would tell us all the answers, but as I just showed you, right, it's more than that. It's also the shape of the chromosome that's important, and who knows what'll be next. And certainly, what are the environmental factors you're associated with, and what is your host genetics? So your DNA is 12 million base pairs different from my DNA. What else is there in those other parts about you compared to me or somebody else that will allow you to do better than somebody else or worse. And these are, all the questions that we're sort of trying to dig through right now.
Of course, thank you. We have some questions as well from some attendees. So one being, patients always ask me if chemotherapy changes their DNA. Can you explain yes or no and why?
Yeah, that's an important question. So now, if you remember, I mentioned that every time your cell divides, you spontaneously will get a mutation or two. Every single cell that divides will do that. Now, when you give, and it depends on the type of chemotherapy, but many chemotherapies will cause damage that will actually induce more mutations. Now, that means as that cell divides, and if the drug, and many drugs will try to break up the DNA so that the cells end up dying, and then cells try to repair that change. When they repair it, a mutation might be introduced. And so that certainly can happen. Luckily, most of the mutations tend to happen in cells that are going to die. So you don't actually see it. But one thing that does happen is, say you had clonal hematopoiesis and you had a small mutation. We know that there are some therapies that will kill a lot of the cells, but the cells that already have the mutation, they're not being generated. If you already have that mutation, that cell is going to be able to survive your therapy. So that cell is going to expand and has potential of either causing a relapse or just expanding so that eventually the disease might regenerate.
Okay, thank you. And then another question here. Would you talk about the germline DDX41 mutation? Why do you think the penetrance is different in males versus females? Also, how do you think a colectomy may impact prognosis or expression of the mutation to start with?
Sorry, did you say a colectomy?
Yes.
So I'll start with the first or the last question first, and I really can't answer that question.
Okay.
I can speculate and hand wave. We know the microbiome. I haven't talked about it at all, but the microbes in your gut. In fact, there's microbes everywhere in your body. Most people are studying the microbes in the gut. We know that has an impact, or there's some evidence that has an impact on clonal hematopoiesis, how those cells grow. How they impact therapy, I mean, we just don't know. So a colectomy could potentially affect your microbiome, which may affect a leukemia, but I really I can't answer that. To go back to the DDX41, so that's a gene that was only discovered to be mutated about 5 to 8 years ago, maybe around 8 years ago, and it's very interesting because, as you say, it was found initially in a few cases, but then as people did more sequencing, they found it to be quite not infrequent in myeloid cancers, and in particular, in many cases, that mutation was actually found to be germline. And for reasons that we don't yet understand, when you have a germline mutation, most people think you'll develop a cancer earlier rather than later because you already have it, very early on. And so there's more chances of acquiring more mutations and then causing the leukemia. But for some reason, with DDX41 mutations, that doesn't happen till later in life. So the median age, so 50% of people who have a germline DDX41 mutation will not end up with a leukemia till they're 62. And, you know, the median age of developing leukemia among the general population is about 68. So it's really not that much different. Why that is, we don't really understand. And the susceptibility in you know, between sexes, there's been some work on that, but it's really not an area that I've spent a lot of time looking at. So I really can't accurately tell you why that is.
Right.
But males tend to have more leukemia than myeloid leukemias than women, and excepting for one kind, which was the deletion of 5q, which I showed you, where women tend to have it more. So
Thank you. And then we have time for 2 more questions. So first one, how long do you estimate this research will translate into treatments for people with AML now, and how soon could this help current patients?
right. So some of the things that I was talking to you about, some of the testing, et cetera, is already implemented. And we're doing it routinely. Others, like the transcriptome, you know, it's just a question of getting it into the clinic and getting funding to do it. And, you know, it's, I used to run that clinical testing environment, but I'm not anymore. So I don't want to really speak for anybody else as to when it will be available, but hopefully in the next year or two. As far as treatments and you know, clinical trials go, you know, the PTK2 inhibitor, as I said, that trial has already started in Australia. And so we'll learn from that. And then the other trial with these new drugs that we hope to bypass resistance of lenalidomide with, we're hoping we can get that started in the next year or two as well. So.
Right. And lastly, we have a question here. What led you or inspired you to do this type of work?
So as a kid, my mom always would tell me about her mom. She lost her mother when she was 12, and being the only girl in a family of 6 kids, she was expected to take on the role of looking after the household. So it was always something that, you know, that's kind of what drove me in the direction of medicine. But then at university in my second summer year, I worked in a lab that did genetic research, but they did it on Drosophila fruit flies. So we're looking at fruit fly DNA, and I looked at it and I thought, here's the future. You know, people weren't doing human testing at the time, but you could actually see DNA, you could do things, you could cut it up, you could take parts of it and replicate it. And I just thought, this is incredible. And I just want to put in a plug for basic research. I mean, that research I was doing was very basic. I was trying to understand how DNA works and how RNA is made from DNA. It was just very, very basic research at the time, with no obvious realization of what things would happen with this. And, you know, there are so many discoveries that are made in worms. We understand how the cells cycle and divide because of incredible research that was actually done in worms. We learned things about how RNA is regulated from plants. So I just, you know, we're all really focused, and I certainly, my lab is really focused on trying to reproduce the human system, because that's what we're interested in. But we can't forget all that basic research that continues to need to be done, so that we can then draw from that and learn how to apply it in a human system to help with diseases. But we shouldn't forget about where those original ideas come from.
Mm-hmm. Thank you so much, Dr. Carson, for being here with us today and helping answer some of our questions we had. If you wouldn't mind flipping the slide for me.
All right.
Thank you. So just finally, thank you everyone for attending today. I would like to remind everyone that we are here for you, so do not hesitate to contact us if you need any more information or support concerning this topic or any of our services. All of our contact information is here on the slide. Please note that also we will send out a short survey to all participants after this webcast. We greatly appreciate you taking a moment to fill it in, as it helps us to offer you the most valuable information in the future. Thanks again, everyone, for attending, and have a wonderful rest of your day or evening. Bye-bye.
Thank you.
Thanks.